First, I would like to thank the organizers for enabling me to talk about our work. I'm actually going to first give a bit of a story of how we got to the early folding prediction in the first place. Now, in structural bioinformatics, typically the amino acid sequence is related to structures, and that information is used to do all kinds of computations. And structures, of course, there's many structures available now in the PDB. And this is a very important resource that has led to many breakthroughs, but it's a static picture. It's the protein in a particular conformation. Whereas, of course, there's a lot of proteins that are quite dynamic, that move a lot, and that adopt a lot of different conformations. And context there is also very important. So, for example, this protein might only really be folded if it encounters the right binding partner. So there's a lot more to proteins and their sequence than the structure itself. One place where you can see that is if you look at the number of sequences in UniProt. There's now over 90 million. And then I used to say that with PDB you have only 30,000 maybe now, so it doesn't even come off the y-axis if you look at this plot. But now with I saw that there's a poster on the SIFTs mapping from the EBI, and they can map about 1.5 million structures now to sequences at the in UniProt. But still, you can appreciate that there's a large amount of sequences for which not much is known. The dark proteome, it's also been coined, I think. So this dark proteome is very interesting, of course, and one thing that you can be reasonably sure of is what's going to be important in this dark proteome are local interactions. So interactions between amino acids that are close to each other in the sequence. The fact that this is important also for structure has already been known for quite a long time, 25 years this year. But in order to really get information about these local contacts and which ones are important, you cannot really use structures because in structures you have a lot of context. There's a lot of residues that are in particular positions that also influence the conformation of other residues. So what we did in our work, we started with this about 5 years ago, is to use data from nuclear magnetic resonance. So in NMR, you put your protein, in solution NMR at least, you put your protein in a sample tube, you record the data for your protein in solution. So it moves, all that information is there. And what you see in NMR is the average over all these different conformations, as long as they switch between them fast enough at least. And one measure that's relatively easy to get, if you can say that, are chemical shifts. So this is a property of atoms in the molecule with NMR. And there's a database called the BMRB, which makes these available. And these are quite easy to get. Now, people have developed a couple of estimators based on chemical shifts that give you the characteristics of residues in a protein, so basically characteristics at residue resolution. The first one was the random coil index by Wishart and Beryansky in Edmonton, which gives you an estimate of the backbone dynamics of that protein, of the residues in a protein, based on the chemical shift data. They also developed a version for sidechain dynamics, and then also work that I was involved in, delta 2D gives you the secondary structure population from the chemical shift data. So these are estimations of what your residue is doing for the protein in solution, more or less. Now, we then use this information to make a predictor. Yeah? So you have a single sequence. We used these estimations to develop a method called Dynamine 4 years ago. So this predicts the backbone dynamics purely from the sequence, a single sequence. And what you get is a profile where, so here on the y-axis, the higher so here's the amino acid sequence, this is the prediction. The higher the value, the more rigid your residue is likely to be. Yeah? Lower values are more flexible, higher values more rigid. This was work done by Elisa Cilia, who was at the ULB at the time, and there's also a web server if you want to try this. Now, So we did the same thing for the other parameters as well. And once we had the predictions, we started to compare it to real data from structures and other sources. And it made us think about what these predictions mean, because they are quite different from methods that are based on structure only. The first thing is that it comes directly from experimental data. So you capture the actual behavior of your protein solution. It might be fully structured, but if a loop is flopping, you'll see that. It also covers a whole range of proteins. This is an important point. So you can record these chemical shifts for IDPs, so fully disordered proteins, to fully folded ones. So you capture a wider range of protein behavior. And finally, we also used for this prediction linear regression. So you capture the overall statistics, the global trends that you see in the data. So in essence, you extract the protein's average behavior in an average context, as it were. So what is the sequence capable of, not what is it going to do in a particular situation. You could also think of this sort of as emergent properties of the protein chain. And one place where these emergent properties of the chain should really come forward is in early folding. So if you have a protein that's completely unfolded and it starts to fold, it's going to be these local interactions that make sure that you get a local structural element that then can influence the rest of the folding. So the idea that certain conformations that this chain adopts are necessary and provide the context for other residues to fold as well. So, and these are formed purely based on local interactions. They don't really need anything else. So basically what we were then trying to find out is, where is your protein primed to form structure? Yeah? So where are these local interactions really telling the sequence that, or make the sequence fold? Now, this data is unfortunately very difficult to get, early folding data. So the first thing we had to do is develop a database called Start to Fold, which is also available, where Michi and Rita, who were in Tompa's lab at the time, went through literature to collect data from NMR and mass spectrometry on hydrogen deuterium exchange experiments that give you information about early folding. We classify them, so early, intermediate, and late folding based on the description in the studies. And we then selected only the early class, so these are the residues that really are the first to form structure from nuclear magnetic resonance pulse labeling experiments. Because there you have residue resolution, but mass spec, it's more peptide fragments. So this data basically is residue resolution, and it detects where your backbone NH becomes solvent protected the first, basically, once this protein starts to fold. And we then compared to the predictions that we had for dynamine, for example. So here you see for whale apomyoglobin, this is the sequence. Here's the backbone rigidity prediction. And yeah, so again, peaks are rigid, troughs are more flexible. And you can see that, I mean, this is qualitative, of course, that the early folding residues, which are in green, correspond in general to peaks in the prediction, which was encouraging, of course. We did a whole analysis on this that I'm not going to get into, but that gave us confidence that we could use this for a predictor. So for this early folding prediction as input features, we want to capture, again, these emergent properties of the protein. And we separate here dynamics from conformation. So we use dynamics prediction, so the backbone and the side chain dynamics, as well as 3 secondary structure classes. So this is not, we don't classify, this is a floating point scale. So we capture both dynamics and conformation, and then use a 5 residue window over the sequence, so that gives us a 25-dimensional vector for the prediction. And these, for this prediction, we only use the predicted propensities, so we don't use amino acid codes, it's really only the predictions that we use, which of course are based on the sequence. So it's used, but indirectly. This data set is very small. There's only 30 proteins that we could use, and then only 27 sets for cross-validation if you take a 25% sequence identity cutoff into account. We use a support vector machine, and then basically what you get is, again, a profile where for a protein sequence you get peaks that tell you where early folding happens, and that basically then would mean that this is where your local interactions really make the structure happen. The performances are not fantastic, but not bad either. So you have about an MCC of about 0.37, I think, and an area under the curve above 0.8 in general in the cross-validation. And this predictor was developed by Daniele Raimondi, who's also at the conference. If you have questions, you can talk to him about that as well. Now, once we had the predictor, then we wanted to check whether it actually makes sense based on existing data. So we first looked at completely independent data from mass spectrometry, at least. A study where they looked at apomaltose binding protein, MBP, and they detected early folding and intermediate folding regions. So what we did is we just predicted the early folding for the sequence, and then we compared first for the early folding for the mass spec, yeah, that's in dark here, and these are the other residues, and this is the predicted early folding. So this is the distribution of the scores for the residues that are in the early folding regions as detected by mass spec, basically. And you can see that there's a highly significant difference here between these two distributions, so that's good, it picks it up. We also corrected for amino acid bias, which I'm not going to get into, but you tend to, of course, have more hydrophobic residues in these regions, so you can try and correct for that. If you look at the intermediate folding regions, you can see that actually there's no difference anymore, which is also quite encouraging because it means that it seems to be able to detect the difference between these two types of residues. And then we also looked at some proteins that are very well studied in terms of folding. One of them is myoglobin. So here you see the structure of myoglobin from N to C terminus, red to blue. And the helix regions here, so this is again the sequence, this is the early folding prediction. The helices are colored according to the structure here. So you have helices from A to H in this case. And in the studies they did of folding of myoglobin, this was the right group, they found that the helices that initiate folding or are the first to fold are A, G, H, and also the C-terminal half of helix B, and that really comes out very nicely out of this prediction. Yeah, so that was also very encouraging. Here's another protein like hemoglobin, which has a very similar structure, so the overall topology is the same. But it was also shown to fold in a different way. See, if you look at the prediction here, you can already appreciate that, yeah, the pattern is very different. And if you then look at the helices that fold first, it corresponds very well with the center of helix E and also helices G and H, but not at all helix A anymore. Yeah? This part they didn't detect in that study, and this is not a helix in this protein. So that corresponds very well with, again, with the experimental studies, which is nice. And we hope that that can help to also design this kind of study. Now, because we train on very generic characteristics, not the actual amino acids themselves, the question is then also, is it useful for IDPs? Because there also you should have some interactions that make the protein fold in particular situations. And if you look at P27, again, here is the sequence, and here is the early folding score. They're in complex with CDK2, P27 forms structure, and here you can see the alpha helical and the beta sheet regions. And in the early folding prediction, these also correspond very nicely to peaks. So this was also a sign that it can work for proteins that do not have any native structure, if you want to call it that. Because the prediction is very fast, we could also go to more proteome scale analysis, so large scale. And we used the Piscis data set with a maximum 20% sequence identity cutoff and X-ray structures of 1.6 angstrom or better, about 3,000 chains. And now what we did is run the prediction on sequences and use a cutoff. If it's above the cutoff, the residue is designated, classified as early folding. If it's below, it's not early folding. And if you look at the relevant solvent accessibility, so that's from the structures, you can see that the early folding residues tend to have a much lower solvent accessibility, which makes sense. but the difference is not that big. The other thing we looked at was the contact S2. This is a measure developed by Bruchweiler, which basically tells you how surrounded the backbone NH is by heavy atoms in the structure. And there you can see that what we predict as early folding residues are really the residues that end up in the final fold as being the ones that are the most surrounded by other residues and other atoms. So this sort of points towards the early folding fragments being really important for the folding process, but then also they remain important in the final structure. So they give the context, as it were, to the other residues. You also compare to Psychov, so I'm going to go a bit quicker because I'm almost out of time. In Psychov, so this is used for contact prediction, right? We picked residues that have at least one covariation signal, so that have some evolutionary conservation of a contact. And you can see that, again, now this is again the early folding scores, the distribution. You can see that if you look at the ones with covariation and without covariation, the residues that do covary tend to also have higher early folding scores, which also makes sense because those are necessary for your fold. In terms of perspectives, we are investigating other things, like, for example, amyloid formation. We use a small data set, REC33, and there you can see that, again, this is the early folding prediction, and these are a couple of residues that we picked from this data set. The lighter shade is amyloid forming, dark is not amyloid forming. You can see that for some amino acids, you really have a very significant difference. For some, you don't really see very much at all, as for tryptophan, for example. So again, it seems to indicate that certain residues are important, other residues are less important, if you think of it in terms of early folding. So what I would like to finish with is that we think that this emergent picture of proteins adds a piece to the puzzle of the whole protein world. So you also need to take into account the more statistical view of proteins that can help, we think, with determining what your protein is going to do. And if you're interested in also working on this, then definitely come and talk to me. I didn't do this by myself, of course. So there's we're in Brussels, so I work together with the ULB, Elisa Cilia, who worked on Dynamite, and Tom Leenaerts, but I'm associated with the VUB, the Flemish one. Daniele developed the predictor. He also has a poster here later. Gabriele has a poster at the 3D6 session, which is related to using this kind of emergent property information for multiple sequence alignment. And Ibrahim is also giving a talk on Monday. And Tashiv was involved in the pictures, and Rita and Mishi with developing the early folding database. Thank you for your attention.
Question for you. Yeah. So you demonstrate there's a correlation between the folding score, initial folding score, and contact number, shortness, SBNT, and other things.
Yes.
Is it possible to incorporate these features into your system to improve the prediction?
Well, but the thing is, we don't want to use any structures. So the whole point is that we stick with sequence, because then you can address all the proteins that do not form structure in the first place.
Yeah. However, some of the features can be predicted, like a second structure, contact number.
This is also true, but in general, the predictors that do that use evolutionary information, which also we want to avoid, because we want to, so all this is single sequence predictions. And then you can do other things with the evolutionary information that you cannot do if you incorporate it in the prediction, we think. So we try and keep it separate that way. Stick with...
Yeah, I have a question. Do you use canonical fragments? Do you use a certain set of fragments, or is it a very large set of fragments? You said you used fragments of length 5?
It's the window over the sequence for the prediction.
Yes, but they are all variable, or is it a defined set of canonical fragments?
It doesn't use canonical fragments. It's a machine learning method, so it just uses the feature, the 25-dimensional feature space to do the prediction.
Ah, okay.
Yeah.
Yeah. Thank you.
Okay. Let's thank Wim for the nice presentation again.